Hello everyone. So today's topic is disinfectant and their role in infection control practices. Myself is Dr. Rohan Dudhale from IMAEC MEDNTEK Limited. So we go through about what kind of disinfectant and exactly their role in the infection control practices. So basically disinfectant is an chemical. So disinfectant describes the process that eliminate many or all pathogenic microorganisms except bacterial spores on inanimate objects. So the process is carried out by using chemical agent to destroy or inactivate or significantly reduce the concentration of pathogenic agent such as bacteria, viruses and fungi. So this is the chemical agent which is known as a disinfectant. So disinfectant and antiseptics are alike in that both are germicidal, but antiseptic are applied to primarily on living tissue, that is known as some skin ointment, skin disinfection purposes. So in which the chemical which is coming in contact with the human skin or living tissue. So it will be depends on what kind of chemicals we are using and depends on what kind of efficacy they prefer to amongst bacteria, viruses or fungi. So there are several sub disinfectant types of disinfectant, which is the application mostly on hand application, hand rubbing, hand sanitizer, skin antiseptic, instrument disinfectant, surface and environmental disinfectant. So basically these are the four main category considered in the disinfectant. Yes, application is several, but the categorization of the hand sanitizer, skin antiseptic, instrument disinfectant, surface and environmental disinfectant play a very important role. So looking at before start with the exactly use of disinfectant, just we try to understand the chain of infection, how infection spread and how to break the chain of infection by using disinfectant. So basically the chain of infection started with the microorganism which is causing infections. So infectious microbes like bacteria, fungi, viruses, prions, protozoas, etc. These are the main bacterial or these are the main microorganism which will be causes the infectious. So that's called as infectious microbe. So what are the first chain of reaction is started with the living things, microbes, microorganism, the reservoir, the place where the microbes lives and replicate, such as peoples, equipments, water, food, animal. So they are present in there, they survive, they grow. After that, the portal of exit, the place where the microbes leaves the reservoir, such as coughing, sneezing, bleeding, fecal matter. For example, if patient is suffering from viral fever, viral diseases, and he suddenly cough and sneeze, if it is not follow the hygiene etiquettes, so what happens by coughing method or sneezing method, whatever inside this bacteria or viruses, they start to leaves their reservoir and they start from the expel from the body. So next mode is mode of transmission. Contact, hands, sharp, injury, airborne, vehicle, insect vector. There are various modes of transmission of disease from one to place. So basically in infection control point of view, they're starting direct or indirect contact. So direct means that is will be involvement of humans by touching, shaking the hands, touching the objects. So inanimate, indirect contact means something, for example, let's say if you are touching continuously on the door knobs or the tabs, in which many people they are coming and touching with without precaution, taking precautions. So that will be the inanimate object who can spread the infection from one person to another person through various routes. So after that, portal of entry points such as wound opening in the skin or mucosa of the mouth via sutures, catheter, IV line. So there is a chance of organism getting entry into the patient body. Maybe there are various ways. For example, if you are not wearing mask, so directly suddenly goes from the nostrils, from the air, suspended air droplets, droplet nuclei especially. This droplet nuclei is very micron in size and they are encapsulated with the just watery droplet inside that there are presence of viruses or some bacteria, so they are transmit from the air current and they are able to like entry into the patients or the patient who are like just a simple measure for putting some IV line. If your the hand hygiene is not performed, whatever the touching objects through the hands, if you are not cleaning the hands and you just inserted the IV line through this medium, direct contact, whatever the microorganisms or infectious agent, they are transmitted from the hands to that medical device through the IV line. So that is called as medical device associated infection. So after a portal of entry, the susceptible host. Who is the susceptible? The organism, once introduced in the human body, they start with their activity to cause the host. Now for the organism, the humans are host, and they are starting with their pathogenic activity. They have several pathogenic islands. So in which the immune system, if is weak or it is not built up in that way, so patient become ill, or they're showing the sign and symptoms about the infectious diseases. So susceptible host is meaning non-immune person, immunodeficiency like babies, elderly, immunocompromised by several drugs like in cancer patient, so much oncological drugs that will be suppressed the human immunity, and what happens, the opportunistic pathogen comes play in picture and they causes the diseases to the human. So it is a continuous cycle. To break this chain of infection, you must intervention of the disinfectant, proper use of disinfectant. It should be very important to break this chain of infection. Now we are talking about the levels of the disinfection. Yes, there are several levels, but basically the three levels which are categorized for the disinfection purpose. One is high level disinfectant or high level disinfection. So can be expected to destroy all form of microorganism with some exception of large number of bacterial spores. Yeah, bacterial spores is very tough and resistance nature for the disinfectant. So that's why killing with the bacterial spore preferably from radiations, ultraviolet radiation, gamma radiation, ETO sterilization. ETO means ethylene dioxide, the chamber in which ETO or gases which are used, which is a very toxic, but they have potential to kill this microorganism. And best is autoclave, because autoclave having 121 degrees Celsius and 15 lbs pressure, and that moist heat, it will be the penetrate to the bacterial spore. So autoclave is a complete form of sterilization, but high level disinfectant which are used, the process may be one step below of the sterilization, that is known as sterilant. So intermediate disinfection, yes, they are inactivate Mycobacterium tuberculosis, vegetative bacteria, most of the viruses, most of the fungi, but does not necessarily kill the spores. According to the hierarchy of the microorganism getting resistance to several chemicals and several antibiotics also. So in this, the chemical which are fall in this intermediate, the chemical or the disinfectant is able to kill Mycobacterium tuberculosis, that is known as tuberculocidal action. And tuberculosis, if you look the cell wall of the tuberculosis with mycolic acid and some secretion of some other molecules, so it is very it is very difficult to kill this Mycobacterium tuberculosis because of the challenging the resistance. So if your disinfectant or a chemical has the power, has the penetration power to kill the Mycobacterium tuberculosis, so that will be the intermediate disinfection. In case if you look at this now upcoming in a hospital, there are drug resistance bacteria, but in the disinfectant subject there is no claim for the MDR. So that's why if your disinfectant is tuberculocidal claim, so that means they can able to kill the multi-drug resistance bacteria. So now talking about the low level of disinfection, they can be able to kill the most bacteria, some viruses and some fungi cannot be relied to on kill on resistance microorganisms such as tuberculosis bacilli or bacteriospores. Yeah, it is a very resistance in nature. So this chemical unable to penetrate, or it is in normal language we can say that it is a penetration power. But if you look at this microbiological aspect, they have able to decrease the bioburden by killing mechanism, so it will be decrease log reduction. So if you look at this level of disinfection by different types of microorganisms, Center for Disease Control given a wonderful picture that how from the starting from less resistance to more resistance, and there is a red arrow in which showing the how it will be the sterilization. So HLD means in yellow arrow, HLD means high level disinfection, LLD means low level disinfection, and in between the high level and low level it will be the intermediate level of disinfection. So look at the heart toughest is mycobacterial spores like Bacillus subtilis, Clostridium species. Below that, this is Mycobacterium tuberculosis or Mycobacterium species. And apart from the microbacteria, there will be small non-lipid, non-enveloped viruses like noroviruses, rhinoviruses, HPV. Then fungus, Cryptococcus pneumoforms, vegetative bacteria like Staphylococcus aureus, E. coli. But most of the, look at, if the Staphylococcus aureus is vegetative bacteria, they can be able to kill with the low level disinfection. But same Staphylococcus aureus converted into like methicillin resistance, Staphylococcus aureus or vancomycin resistance, enterococci, then you have to need intermediate level of disinfectant. So below the vegetative is some lipid enveloped viruses like SARS, influenza virus type A. So they require low level like alcohol. They can be able to kill this microorganism by this hierarchy. These are the levels of disinfection by the types of microorganism. So going for the another aspect for the classification of these medical devices by Dr. Spondling has given wonderful idea about the what kind of material, what kind of approach to the patient contact, and what kind of disinfectant level should be used. So they are categorized non-critical, semi-critical, and critical. First, try to understand what is a non-critical, semi-critical, and critical medical devices. First, the non-critical probes contact intact skin. Intact skin means there is no broken skin. So intact skin like a stethoscope or a blood pressure apparatus that will be useful in the on intact skin. So there is a need only low level of disinfection. So look at semi-critical medical devices. So that will be the probes contacts with non-intact skin like wound, burns, and mucous membrane. For example, like your endoscope directly touching with the blood and mucosal line. So that will be the very semi-critical medical devices, but it need a high level disinfection. For the critical, probes contact with sterile tissue, body fluids, blood stream. So it will be the various surface ultrasound, nerve blocks, biopsies, central line placement, peripheral IV access, because they're directly involved to the sterile or directly blood stream or any touching the sterile tissue. So it should be ideally it should be a sterilization, or if the whatever the devices or medical devices it is a very heat sensitive, then it should be goes for the high level disinfection. Now we are looking the chart of various chemicals which are present and categorization and their efficacy, local human toxicity, and application. First look it up, we categorize these disinfectant in alcohol, chlorhexidine, chlorine compound, formaldehyde, glutaraldehyde, hydrogen peroxide, iodophores, peracetic acid, phenolic compounds, and quaternary ammonium compound. So starting with this alcohol, this chemical group, I just explain with this alcohol. Look at this. Alcohol has showing the efficacy. Majorly only two form of alcohol they can use. For example, ethyl alcohol and isopropyl alcohol. Due to the methanol is a very good antimicrobial efficacy, but due to the toxicity is very high, so that will be not useful in the disinfectant purpose. So bactericidal, if you look at this claim, bactericidal means you have the chemical is able to kill the microorganism. So cidals mean lethal effect or killing effect. So bactericidal means they are able to kill the bacteria. Tuberculosidal, yes, they are able to kill the Mycobacterium tuberculosis or any other Mycobacterium form. Fungicidal, yes, it will be killing the fungus. Virucidal, it is against the viruses. And sporicidal means again bacterial spores and various fungal spores also. If you look at it, local toxicity, yes, when you are handling the disinfection, there will be exposure to the humans. So that's why it will be as a good ideal disinfection, it should be a low to moderate risk of the chemicals. And application, alcohol mostly used as skin antiseptic disinfection and hand rubs, and even including some small surface area to clean and like wipes and some disinfectant. So look at the chlorhexidine also, they have some very weak molecule, but along with chlorhexidine and alcohol it will be boosting the effect of antimicrobial compound. Same way chlorine compounds. Formaldehyde, yes, formaldehyde is a very active molecule, but it is toxicity high, and now the OSHA guideline, occupational safety hazards, so they are under classified as a potential carcinogen. So look at the glutaraldehyde and orthophthalaldehyde, which is mostly both are the aldehyde groups. Glutaraldehyde is also very high toxicity chemical, but efficacy is very high against all forms of bacteria, like including spore also. So but the toxicity has high, so that will be the, when you are handling the glutaraldehyde, due to this exposure level, to minimize this exposure level, you have to wear the personal protection, personal protective equipments, PPE kits. So if you had a look at this hydrogen peroxide, most of the hydrogen peroxide which are used for the wound, like let's say 1% to 6% they are using, but newer combination like accelerated hydrogen peroxide with stabilized formulations, and these hydrogen peroxide along with some stabilizing molecule like silver, so they can helpful for this surface and environmental disinfection. So iodophore, yes, iodophore including iodine, so it will be the moderate to the skin, so that's more preferred for the skin and wound dressings. Mostly povidone iodine is which are used like 5%, 7.5% and 10% for the pre-operative procedure. Generally surgical wash or surgical scrubbing, povidone iodine is a best choice for the surgeon. Now it is peracetic acid. Peracetic acids is also being used. Phenolic compound, nowadays phenolic, most of the bacteria like pseudomonas which are mostly resistant to this phenolic compound, but depends on what kind of exposure. In hospital, yes, there are maybe potential infectious agent, but in household mostly phenolic compound which are used like in general disinfection purposes. Quaternary ammonium compound, now this is upcoming and eco-friendly molecule like quaternary quartz, generally called as quartz. They have several generation, first, second, third, fourth and fifth, and latest 6 7s also they are upcoming. But why the generation is need? In generation means there's updated version of this compound in which there is changing the side chain with a various group, and it boosts the antimicrobial efficacy as compared to first generation. Second generation is quietly high efficacy towards the microorganism. So fifth generation is also how it will be made generation. It is nothing but the combination of these first and third, like for example fifth generation quaternary ammonium compound made up of fourth generation and second generation of quaternary ammonium compound. So it will give good combination results of the quaternary ammonium compound, and it is mostly, it is not a toxic form, it is a very low in toxicity, and it is a biodegradable molecule. So how does disinfectant work? If you look at this various in this animation, Mr. Jam is here, make you see sick. So spraying Mr. Jam with the disinfectant start to break down his cell wall. So depending, that is known as mode of action of various chemicals. If you know in the like drugs, there is a specific mode of action. So disinfectant also plays in such a way that they are specific to like denature the protein, denature this cell wall structure, destruction of cell wall, even rupture of the membrane, rupture of the bacterial spore. So this disinfection dwell times break down more of Mr. Germ wall. So germ, it will be break this microbial cell wall. So automatically the Mr. Germ will be die. So with his time and the concentration of disinfectant, it will be potent to the microorganism. So there is various definition: contact time, dwell time, kill time. The time a disinfectant is directly contact with the surface or item to be disinfectant. So dwell times vary by disinfectant and target organism type like bacteria, viruses or fungi. They ranges from 30 seconds to 10 minutes. For example, just we have this intermediate disinfectant. They can generally kill the microorganisms, general microorganisms 10 minutes. But if it is a Mycobacterium tuberculosis, it take 30 minutes. So this is a variation. Molecule is same, concentration is a different, or maybe concentration is same, but contact time is dual. So concentration and dosage, the efficacy of the disinfectant is depend on the concentration of the disinfectant. Yes, it is exactly perfectly. If you say that the concentration play major important role. You know, in microbiological aspect, or when you study with the antibiotics, so minimal inhibitory concentration or dosage which required, they are able to kill the half of the populations of the microorganism. So that will be the minimal inhibitory concentration. So concentration is very important. Acute concentration along with the time is very important in the disinfectant. So now we are looking about these chemical groups, how it will be started with nature of this molecule. Alcohol based disinfectant, mode of action is denaturation of the proteins. Protein denaturation also is consist with the observation that alcohol destroy the dehydrogenase of the E. coli and that the ethyl alcohol increases the lag phase of enterovector erogens, and that the lag phase effect could be reversed by adding certain amino acid. So organism able to, some of the most of the organism like spores are resistant to alcohol. Alcohol is unable to penetrate the bacteriospore, but some viruses and bacteria they are able to, definitely they are able to denature the protein. So absolute alcohol could not work when you adding the water. So minimal inhibitory concentration of the alcohol is according to WHO and CDC guidelines 60 to 85% of the concentration of alcohol will be the germicidal range, but best actively, for example, like best like percentage, if you look at 70% is ideal for the of the alcohol to give the best logarithm log reduction in the microorganism. So based on this alcohol based disinfection, we have two products which is the WHO recommended, like ProVibes Sidaltech IP and Sidaltech EN. These are the two products which have the alcohol based disinfectant. Both are the IP means isopropanol and EN means ethanol. So these two formulation given by WHO, World Health Organization. So there are several alcohol related problems come because continuous exposure to alcohol may be skin irritate. Alcohol has the nature of removing the water. The skin become dry, and when dry skin, the skin get rupture and bacterial contamination or fungal contamination started, and it's various skin diseases like skin dermatitis problem associated with the most user of the alcohols. So there will be having non-alcohol based disinfectant. Yes, benzothonium is a synthetic quaternary ammonium salt which is a first generation with surfactant, antiseptic and broad spectrum antimicrobial properties. So most of the useful of this non-alcohol based like benzothonium skin disinfectant at the concentration of zero point 0.1 to 0.2 percentage, which is a safe and effective concentration for the compound specified by the United States Food and Drug Administration. So if you look at the structure of this benzothonium chloride, is a first generation quaternary amine compound. It is a microbial and it is falls under the low level disinfection. So microbial activity and obviously the local toxicity to the humans is very low. So that's why it will be using for the hand sanitizer and various cosmetic purposes also. Maybe they are used for the preservative also, this molecule. So microbial action, it is shown to be the effective against bacteria, fungi, mold, viruses. There is evidence that benzothonium act as a spermatocide but may have the vaginal irritation, causes vaginal irritation. Benzothonium was identified as a novel cancer specific compound by cell based small molecule skin. Obviously this nature they can useful for the another diagnostic purposes in cancer treatment also, but due to this microbial efficacy, so benzothonium and another is benzalkonium, both are the first generation quaternary amine compound which is falls under non-alcohol based disinfectant. Now we go looking about the glutaraldehyde. Glutaraldehyde is a very high level disinfectant and which is also known as chemical stirlant. Only when the solution is activated male alkaline by using alkalizing agent to pH 7.5 to 8.5 does this solution become a sporicidal. So when you see the glutaraldehyde which is remains not activated, so there is useful the chelating agent like activator. When you adding this activator, the solution become sporicidal. The solution drop the pH, so it will be more towards the alkaline, and then the solution is known as active form. Once activated, this solution has a shelf life minimum 15 days. So mode of action, this how glutaraldehyde acts. The biocidal activity result from its alkylation of hydroxyl, carboxyl and amino groups of the microorganism, which can alter RNA, DNA and protein synthesis. If it is with the alteration that is known as mutation, so apoptosis it will be happening because it is alteration of DNA bases that will be leads to the unrecognition reading for this microorganism, and it will be disrupt the protein synthesis that leads to the cell die, the microbial cell will die. So it will be used high level disinfectant for medical equipment like endoscope, spirometry, tubings, dialyzer, transducer, anesthesia, and respiratory therapy equipment, in hemodialysis or dialysis delivery system, or any reusable laparoscopic disposable plastic tarot cards. So basically mostly the medical device which is unable to send process for CSSD like autoclave. So it will be heat sensitive medical semi-critical medical devices. So glutaraldehyde they can use. So glutaraldehyde has having good material compatibility, but colitis is believed to be caused by the glutaraldehyde residual exposure in the endoscope solution channel. If you disinfectant with the glutaraldehyde solution, and after that if it's not properly washed with the water, sterile water, what happen? Some residues of glutaraldehyde, it will be interact with the patient mucosal membrane and it will generate the colitis. That is known as glutaraldehyde associated colitis. So if you look at its various cells target for the biocidal agents, there if you look at chlorhexidine, glutaraldehyde, peracetic acid, phenol, phenolic compound, they will be the coagulation of cytoplasmic content. So they will be disturb this cytoplasmic content. So various activities of this molecule which will be specified. For example, if DNA is glutaraldehyde, chlorhexidine, formaldehyde, hydrogen peroxide, iodophores, they will be alter the DNA sequences. If you look at the hydrogen peroxide, they directly impact on microbial ribosome. So if you look at the cell wall like aldehyde, halogen, peracetic acid, phenolic compound, they will be disturb the cell membrane or break the microbial cell membrane. Now we are going for the orthophthalaldehyde. Orthophthalaldehyde, which is known as OPA, is again high level disinfectant. It is an aldehyde group that received the FDA clearance in October 1999. This molecule is well accepted by surgeons for the disinfection of the endoscope. So it will be used. It will be used as a high level disinfectant. So mode of action, the OPA and glutaraldehyde interact with the same because both are the aldehyde group, interact with the amino acid proteins and in the microorganism. OPA appears to kill spore by blocking the spore germination process. So mostly OPA is not showing the sporicidal activity, but in within time it takes many like 3 hours or more than 3 hours it's able to kill. But OPA has instant action, small contact time, they able to kill the Mycobacterium tuberculosis and other bacterial and viruses. So it has excellent stability over wide range of pH. It should be stable, safe to the eyes and nasal passage, does not require any exposure monitor, has barely perceptible odor, and there is no requirement of activation. OPA like glutaraldehyde has a excellent material compatibility. So IMAEC MEDNTEK Limited having this Cydeltech OPA is our product. So several studies showing that the OPS has a very superior Mycobacterium sidle. Within 5 minutes they can decrease the as much 5 log to the base 10 reduction as compared to glutaraldehyde. So OPS is also showing good activity against the microorganism tested against the glutaraldehyde resistance Mycobacterium strain. Most of the Mycobacterium tuberculosis strain, they are able to resist the glutaraldehyde, but OPA which will show the good activity against glutaraldehyde resistance Mycobacterium strains. So now going for the quaternary ammonium compounds. Quaternary ammonium compounds are widely used as a disinfectant since from 1930s. So if you look at the structure of quaternary ammonium compounds, so the quatern means 4R group, functional group, and they have starting with the side chain. So the quats are very good cleansing agents. So the property of the good cleansing agent. So that's why in any household disinfectant or any disinfectant having with the quaternary ammonium compound, it will be act as a good disinfectant along with good cleaning agent. So mode of action of this quaternary ammonium compound, this is the bactericidal action of the quats have been attributed to the inactivation of energy producing enzymes, denaturation of the essential cell proteins, and disruption of the cell membrane. So basically mostly useful in the household surface and environmental sanitization, environmental disinfectant they can use. And this molecule is Environmental Protection Agency, it is a register because it is a biodegradable molecule. It is not toxic to the nature. So it will be started from this generation. So quartz has been sold as a hospital disinfectant, generally fungicidal, bactericidal, virucidal, tuberculosidal. So these are the generation. If you're talking about the microbacteria like quaternary amine compound, they will be having various generation. I already mentioned and already told regarding this. How it will be generation is a combination of uh, first to second or fourth to and second. It's for, the mixing of this quaternary compound. There is a mixture of fourth generation quaternary amine compound and second generation of quaternary compound. It become a fifth generation. Now we are looking after the hydrogen peroxide. So it is a oxidizing agent. So H2O2, 2H and 2 oxygens. Hydrogen, 2 hydrogen and 2 oxygen molecules. So hydrogen peroxide works by producing destructive hydroxyl free radicals that can attack membrane, lipid, DNA and other essential cell components. It will be usually 3% of the hydrogen peroxide is a stable and effective disinfectant when it is used on inanimate surface. So hydrogen peroxide is effective in disinfecting fabric in patient rooms. So it is will be a very useful and it will be disinfectant. It has been used as a concentration from 3 to 6% for disinfecting soft contact lenses, like 3% for 2 to 3 hours, tonometer, biprism, ventilator, fabric and endoscope. So microbial to release the free active molecules, free radical. So hydrogen peroxide is a very active against the wide range of microorganisms including bacteria, yeast, fungi, viruses, spores. A 0.5% accelerated hydrogen peroxide demonstrated bactericidal and virucidal activity within a 1 minute and mycobactericidal and fungicidal activity within a 5 minutes. So this is a hydrogen peroxide. Look at it, the pictures, how it will be help acting on the bacteria, on the bacteria. So our product is Cydeltech W10 is a the disinfectant which will be the play important role for the controlling the surface and environmental disinfection. So now we are going what kind of factors will be affecting on the efficacy of the disinfection. There are several factors. First we go through the innate resistance of microorganism. If you are using disinfectant, microorganism, if you not properly follow the contact time and concentration, what happened, the microorganism started getting resistance mechanism in end of the way. They are secreting some polysaccharide secretion, they are blocking the entry of that particular disinfectant, maybe the alteration of this molecule, efflux pumps they have starting, or maybe by genetically taking the plasmid with the help of plasmids, so they can create a resistance genes towards this disinfectant. Now concentration and potencies of the disinfectant, if you not read the disinfectant label properly, then the unproperly means an improper use of this concentration, that will be affect on the killing mechanism of this particular disinfectant, and if you are not following the contact time, then it will be impact. So physical and chemical factor, yes, most of the sunlight and the change in alternation of pH, it will be impact on the disinfectant product efficacy. Number and location of microorganism, yes, it will be the, say, let's see if you have this disinfectant, they can able to kill several microorganisms, but if you don't know about how the surface is associated with this microorganism, so that means you need to take the more concentration along with the contact time proper. So that will be the factor that will be effect on the disinfection process. And location, yes, if it is disinfecting like tubings, so tubings we don't know how it will be, not the horizontal pathway, horizontal surfaces, so due to the gaps, the unable disinfectant will be reached, and this whatever the device, it's again getting contaminated, and maybe this small portion of disinfectant come contact, this microorganism able to develop the resistance. Organic inorganic matter, yes, it is very important, in which most of the like dirt, physical dirt, they can interfere with the efficacy disinfection. That's why like a 2 bucket and 3 bucket system for cleaning is a very crucial role and mandatory to avoid the disinfectant getting contaminated and to reduce this disinfection efficacy. So to remove first organic and inorganic matter, for example, if you are doing the endoscope cleaning disinfection, first you have to clean to physical removing of the all physical like blood, mucous membrane, they can interfere with this disinfection efficacy. So duration of exposure, yes, proper contact time under wet condition it should be given. Suppose let's see, it is a disinfectant wipes, it required 10 minutes of contact time, and if you are immediately you are touching with the surface, there is no use of using disinfectant, because you are adding extra microbial load, which is, so proper contact time should be followed. And biofilm, yes, most of the organisms that develop a biofilms. You know, biofilms means single stick is able to break, but bunch of stick is required more power. Same thing like in bacterial, they form a huge number of bacteria comes together, form a very protective secretions like polysaccharides, so protecting layer, so that will be the disinfectant unable to penetrate. So that will be, these are the several factors which can be affecting the efficacy of the disinfectant. What is the properties of the ideal disinfectant? What is the ideal disinfectant? It should be broad spectrum. Yes, it will be acted bacteria, yeast, fungus, viruses, spores. So it will be broad spectrum, fast acting. Yes, as soon as contact with the surfaces or contact with the bacteria, they start with the activity in a rapid manners. Not affected by the environmental factors. It should be a stable, non-toxic. Yes, it is a very important when you handling such kind of disinfectant. It is should not be toxic to the by the end users. Surface compatibility, yes, most of the surface like polyvinyl, vinyl, stainless steel, copper, by using this disinfectant they could not damage this material, so that will be the material compatibility issues will become. So surface compatibility should be good. Residual effect on treated surface, yes, once you treat the clean or disinfectant the surface, to some extent it should showing the antimicrobial activity on the treated surface. So while touching with the bare hands of the treated surface, it should be remain disinfectant by using their residual activity. Easy to use with a clear label instruction. Yes, it should not be create chaos to the end users. But after reading the disinfectant labels, it should be clear cut mentioning that what is the concentration or what is the dilutions, how much ml of disinfectant mixed with how much ml of water, and how much it should be follow the contact time, and by the disinfectant delivery method, it should be spray or fogger or just wipe. So it will be a very clear instruction. Odorless, yes, it should be odorless. Why? Because the disinfectant we are adding with some fragrance to create like a perfume, like a flower like lilies and strawberry flavor and mango flavor, or citrus lemon flavor. So what happens when you adding such kind of chemicals? So it will be starting with the irritation problem, maybe leads to the asthma or hypersensitivity issue. So that's why, so typical odorless should be the chemical should be used, and it should be economical. Solubility, yes, properly soluble it is. Like most of this problem associated with the solid disinfectant. Yes, powder form of disinfectant, when you are adding with the water, they remain suspended or either they are stagnant. So it should be mixing or soluble in whatever the mixing in water. Some of the disinfectant were showing some like suspended like oily. So it should not be. It is a proper mixing. It's soluble of the disinfectant. Stability, yes, over period of the shelf life of that water, the label, it should be stable. It should not showing any kind of reactions. It should be good cleaner. Yes, it should be good cleaner and environmental friendly. Yes, it should not damage to the environment. So these are the several points for the ideal disinfectant. So when you consider selecting, many factors must be considered when you selecting the disinfectant. Yes, the first is kill claims. What is your applications? First you have to decide key, just you wanted to what kind of your application, maybe surface, maybe environment, maybe skin, maybe hand. So just find out what you want for the using disinfectant. Then disinfectant activity claims. Yes, if you the patient having some tuberculosis, what obviously I need a disinfectant which can able to kill the mostly tuberculosis or drug resistance tuberculosis. So tuberculosis is a very important claim in that using particular disinfectant. Easy to use, like contact time, correction, correct concentration or dilutions, mixing requirement. Yes, external mixing requirement, they should be required some water water, some additional requirement. Stability, yes, product should be very stable. Need a separate cleaning step, yes, it should be defined how it should be cleaned with this disinfectant. Method of delivery, yes, it should be taking the spray form, fogger form, 2 bucket, 3 bucket system. Safety concern, yes, material safety data sheet along with this product is usually provided. So toxicity and flammability, it should be very precisely. So end users must be trained, their colleagues, their other healthcare workers while handling this disinfectant. What kind of PPE should be considered, how it will be affecting if any goes to the eyes or inhale. So first aid, it should be immediately informed. So surface compactability, persistence, activity and odor, these are the several points must be considered when you selecting the disinfectant. So MEDNTEK Limited having the Tech under brand name. These are the various skin disinfectant and hand disinfectant, instrument and surface. So we have this disinfectant like hand sanitizer. We have two alcohol based hand sanitizer and one is non-alcohol based hand sanitizer. Then instrument, we have two product, one is aldehyde containing disinfectant and one is aldehyde free. We have the three environmental and surface disinfectant product based on hydrogen peroxide, quaternary ammonium compound and triamine technology products. So if in case of any query, questions, or if you need to want this disinfectant from the IMAEC point of sight, so please communicate it with this following contact number, and we have this customer support also. You can visit our website, social media page like Instagram, Facebook, YouTube and LinkedIn. So once again, I'm very much thankful. If any case of any doubt, please communicate to IMAEC MEDNTEK Limited. Thank you.